7-phosphate. So the question is, which carbons of these are going to be labeled, first labeled when you add the C14 to the chloroplast? So this is a classic experiment, just as Kelvin carried it out. And what you need to do here is use this diagram that is up here. And again, it's a little difficult to see in the room here, but if you print it out or look at it on your computer, it'll be fairly clear. So what occurs here then is we add the C14 on the CO2 there. It ends up on that carbon right there of the 3-phosphoglycerate, and then it becomes part of the 1,3-bisphosphoglycerate, glyceraldehyde 3-phosphate. It ends up on those, and again I can hardly see it myself here, but it ends up on those middle two carbons of the fructose 6-phosphate. So it ends up at that carbon there, and that carbon there, and that carbon there, and that carbon there. When this is converted into the erythrose, that radioactive carbon is on the top two carbons of the erythrose phosphate. When that reacts with the dihydroxyacetone phosphate, which has the third carbon labeled, it ends up on carbons 3, 4, and 5 of this cediheptalose 7-phosphate. So the answer is carbons 3, 4, and 5. So where the radioactivity is going to occur here is going to be this carbon here, this carbon there, and that carbon there. And so you follow this through the pattern, just following the color scheme on this Kelvin cycle. And I would encourage you, you can kind of figure out, there's some other subsequent labeling patterns where they show how the different carbon fragments rearrange as you go through these transketolase and transaldolase reactions. So I would recommend that you just study this a little bit and compare it with some other diagrams, and you can follow how the carbons are moving through this carbon cycle and how they're rearranging as a result of all these transketolase and aldolase reactions. Okay, any questions on that? Okay, one other question that we had on this exam that relates to photosynthesis is going to be question 20. So let's take a look at that. So this question 20, we have a thylakoid membrane. I'll just draw it like this. And the pH is 6 on the inside of the membrane, and the pH is 9 on the outside of the membrane. Remember that during photosynthesis, the electron transfer in the Q cycle pumps protons from the outside to the inside, so there's a higher concentration of protons on the inside of the membrane. We then also specify that there is a voltage across the membrane, which is going to be +1 on the inside, plus 0.1 volts on the inside, relative to 0 volts on the outside. And then the question is to calculate the delta G for transferring 1 mole of protons from the outside to the inside. So that's the question. And we had a formula for this when we went through the mitochondrial respiratory chain that's identical to the formula for proton transfer that you use during the photosynthesis. And that formula is going to be equal to RT logarithm of the proton on the inside divided by the proton on the outside. You've seen that type of equation before, plus Faraday's constant times the voltage on the inside minus the voltage on the outside. And so this is now simply converted into using your numbers that are given, that will be given on the exam, and were given on this exam, and that you should also have in your notes. So this is 5.7 log to the base 10 of the protons on the inside divided by the protons on the outside. And Faraday's constant is 96.5. And this is going, I'll just put in the value here. This is plus 0.1 volts. And so all of this now is going to convert into 26.75 kilojoules per mole. So this type of calculation for the energy production in the chloroplast is pretty much identical to what you had for the mitochondrial respiratory chain, and you use a formula like this to get these, this value. So you get quite a bit of energy out of just pumping one proton towards the inside of the membrane. Notice that this is positive, means that it's unfavorable to pump that proton, and that is coupled to the electron transfer process in the Q cycle for that reaction to occur. Okay, so that's an example of another type of question that we could have on the photosynthesis. There's a number of others that we could have as well. Any questions on that?
Will you have that free energy calculation, or?
Pardon?
Will you have that free, find the free energy or the, never mind.
Did you have the formula, you mean, or?
Will you have a question about calculating the free energy, or?
Well, this is calculating the free
Okay.
energy, so this is an example of that type of question. But it'll be limited to something involving photosynthesis or DNA, if there's anything involving DNA reactions or, you know, protein synthesis, something like that, we could have a free energy question. Okay, well, let's switch now and go on to the to the DNA types of questions. And I'm going to switch to exam 2 from 2011. And let me switch here. And there's a number of questions on the old exam that involve mutations and the use of the genetic code. And so you can see the consequences of mutations in DNA and how they affect the translation into specific amino acids in a protein. So let's go here. I won't start at the, well, I think maybe that fourth one is going to be a good one to look at. So let me, if you have this exam with you, or otherwise I'll write it up here.
Bless you.
This is exam 2 of 2011. And let's first of all look at question 4. And what question 4 asks is, what process is least affected by a mutation that inactivates the pyrophosphatase? This is the enzyme that carries out this reaction here. Pyrophosphate plus water goes to 2 molecules of phosphate. So you have a mutation that kills this enzyme. This is carried out by the enzyme called pyrophosphatase. So we have a mutation that kills this enzyme. And what is that going to affect? And I give a whole lot of different possibilities here, but let's just talk about those in general. Remember that when we talk about DNA replication, transcription, translation, we start with replication. And remember that the reaction that is carried out there is that we take one of the 4 nucleotide triphosphates. Let's just say that we're using the D-ATP, for example, as one of those things. That is then incorporated into DNA. So the ATP is incorporated into DNA. We form the new phosphodiester bond, and we release pyrophosphate, and that then is hydrolyzed to 2 molecules of phosphate in the presence of water. So that is the enzyme that is carrying this reaction out. So each step of replication, each new nucleotide we add, the energy to drive this reaction comes from the hydrolysis of pyrophosphate to 2 phosphates. We use this energy of ATP twice, in a sense. We first use it to form the new DNA molecule. Let me expand on this. This doesn't look quite right here, quite complete, I should say. We take the ATP plus a DNA with N molecules, and then we add the DA to the end here, and then we have that attached to the DNA of N molecules, so now we have N plus molecules, and this releases the pyrophosphate, and that is hydrolyzed to the 2 phosphate molecules in the presence of water. So every nucleotide that we add to the DNA during the replication process requires this enzyme to provide the driving force. Without this enzyme, this has no driving force, and it simply isn't going to occur very rapidly. So the pyrophosphatase enzyme is what provides that reaction with the driving force, and it allows it to go rapidly. So replication would be affected dramatically by that mutation. It would probably stop or slow down to a very slow value. What about transcription? What type of mechanism do we have there? It's the same thing. We use one of the activated nucleotide triphosphates, in this case the regular ATP or UTP or GTP, etc. We form the new phosphodiester bond, growing in the 5' to 3' direction, and then the same thing happens. We release the pyrophosphate, and that's hydrolyzed. Again, that reaction provides the driving force of the reaction. So transcription would be greatly affected by the mutation that inactivates that particular enzyme. The process that would not be affected is the synthesis of proteins on the ribosome, because that uses 2 molecules of GTP, which are used in 2 different steps of that translation process on the ribosome. It does not directly involve the formation of pyrophosphate. So the only answer that is correct here in this question 4 is going to be D, protein synthesis on the ribosome, assuming that the messenger RNA and all of the amino acid transfer RNAs are available. So the answer here is D, and it's only the direct reaction on the ribosome that does not involve the formation of this pyrophosphate. Okay? So all the other reactions we talked about with DNA involve that pyrophosphate. So that's a critical part of the energetics of the DNA replication and transcription. Okay, any questions on that? Okay, let's do one of these questions about how many high energy bonds. This will be question number 6 of this same exam. So, how many high energy bonds? And what we're talking about here now is we have a bacterial gene that codes for 700 amino acids. But what I want to know is how many high energy bonds are needed to replicate the DNA that codes for that gene. So let's put it all down here. Here is the DNA for that gene. That is now going to be transcribed into a messenger RNA of the same length. Let's just suppose this is the gene right here from there to there. There's no introns in bacteria, so we don't need to worry about that complication. This then is going to be translated into an amino acid, starting with the amino terminus and the carboxy terminus. And we say that we have 700 amino acids. Okay? So, how many nucleotides do we have on the messenger RNA for the molecule? If we have 700 amino acids, how many nucleotides do we need on the messenger RNA? Okay, it's going to be 3 times that, because each codon of 3 nucleotides codes for 1 amino acid. So if we have a protein with 700 amino acids, we have a messenger RNA with 2,100 nucleotides, okay? And how many molecules of nucleotides do we have then in the original DNA gene? Well, we have a coding strand and a template strand, so we have 4,200 nucleotides in this gene, right? So we just double those. Now the question is, when this is replicated, how many high energy bonds do we need? And during the replication, we are going to make 2 new strands, 2 daughter strands, and so we're going to double the amount. So we're going to replicate a new piece here and a new piece there to make those daughter strands. And so there's 2 new strands that are used to carry that out. And for each one of these nucleotides that are attached, we need how many high energy bonds? We just talked about this over here. We need 2 high energy bonds, because for each one of these, we take a nucleotide like the ATP. We use one of the high energy bonds when we take the DNA and attach the A to that. And then we produce, as part of that, the pyrophosphate, and that is converted into 2 phosphates with a molecule of water. So one of the bonds is used to make the new bond, and the other bond is broken when the pyrophosphate is broken. So the total number that we have here is going to be 700 amino acids times 3 to get us the number of nucleotides in the messenger RNA, times 2 to give us the number of new nucleotides we have to synthesize on these 2 daughter strands, plus 2 more, or times 2 more, and that's the amount of high energy bonds for each nucleotide that is added. And so the total number here is 8,400. So that's the final answer. So one thing I emphasize a lot here, again, is what I call metabolic accounting. And this is an example of metabolic accounting, figuring out how many high energy bonds are needed to carry out a certain metabolic process like replication. Replication is very energy intensive, as you can see by the numbers here, 8,400. All of that has to come from the mitochondrial respiratory chain and the citric acid cycle and all of those types of metabolic processes. So that's what we need to do there. Any questions on that?
I have a question.
Yeah.
After we go from 4,200, which is just the total 2 of the separate nucleotides,
Yeah.
the next 4,200, do I multiply it by 2 again because there are 2 daughter strands? Is that why?
No, once we got up to 4,200, we have the 2 strands. Each of these strands is replicated with a new daughter strand, but we only, I should have a color coding here, but let me put the parents, I'll put as X. So here are the parents, and these are the new strands. So the number of nucleotides is still 4,200. The last factor of 2 comes because we need to break 2 high-energy bonds for each nucleotide that is added. We start out with an ATP. We break 1 bond to attach it to the growing DNA. We make pyrophosphate, then we break that apart as well. So that's why we get the 2 bonds. Okay. Question 7 is related to that, but I won't go through that one. Why don't we do this number 8? And I got that genetic code up on the board there, and we might as well go ahead and use that. So there's a lot of questions that you can have about how you do these mutations and what effect they have on a protein that's going to be synthesized. So let's look at question 8 here. And we have a protein that has 150 amino acids, 1 to 150. We then say here that we have a single point mutation. So we make a new protein that has a point mutation here. And this is going to be called mutant A. So this is mutant A. And it's identical to the original protein, except at residue 53, it becomes a threonine. So here, this is a threonine. It started out as an arginine. So we're making a point mutation at residue 53 from arginine to threonine, okay? Then we have another separate mutation. I better put that over here. And this is called mutant B. And this is starting with the original molecule, but now we have that same residue 53, which is now changed into a tryptophan. So this is tryptophan at residue 53. So here we change arginine into tryptophan. So this is mutant B, this is mutant A here. And the question then, what is the codon for the original amino acid in the wild type enzyme that's coding for arginine? And what does it change to in the threonine and in the tryptophan in mutant B? And the way we can work that out is that we have to look up here at the genetic table. So we start out with arginine. And you can see we got 6 different possibilities, right? There's 4 codons up there for arginine, 2 more down there. Our first mutation is to a threonine. Well, I think you can see it's got to come over here, but it could either be AGA goes to ACA or AGG to ACG. So either one of those is possible. We can rule out all of these guys by that mutant A. We can't get from any one of those to a threonine by a point mutation. Remember, the rule here is that a point mutation, you have to move horizontally or vertically, and then there's even limits on that. A point mutation is only 1 nucleotide, okay? So that first thing says we have to be one of those 2 going to one of those 2, but we don't know which. Now, mutant B, we start with the arginine, one of those 2, and we go to the tryptophan. Well, this is where it's easy. There's only 1 codon for tryptophan. You see that? So, there's no choice here. We have to go from AGG to UGG. We could have gone from CGG to UGG, but that position is not consistent with getting to threonine with a point mutation. So, the answer here then is going to be, if we look at, where am I? The answer here is going to be D. So this is going to be AGG on the original. the threonine is going to be ACG for the codon, one chain, G to C. And then the tryptophan, this other one, is going to be UGG. Again, a single change, AGG to UGG. Okay? So that's how you can work out these mutation problems. And again, the geneticists really have gone wild with this, and there's lots of different possibilities. One thing I would encourage you to do with these questions is read the question very carefully, because I have a habit of making a question that looks very similar, but changing it around a little bit. So read it very carefully. Another type of question would be that if I took the original mutant to the original wild type to a new mutant, and then use that mutant as the starting point to make a second mutant, okay? So there's different ways in which these can be done. So be sure to follow that carefully. Let me let me give question 9, the very next one, because I think that illustrates another way of doing this. So what we have in question 9 is we have 140 amino acids here. So here is our 140, starting at 1 here. And at residue number 57, we have an arginine. And we go into a mutant, and we generate in that mutant at that same position a inactive protein. So we have, so this would give a protein with an arginine, something coding for an arginine. This would give a protein now that stops right there. So it's an incomplete protein of only 56 amino acids, with the amino terminus there and the carboxy terminus there. What's wrong? Why do we get that incomplete protein? Well, the only thing that could happen here is that it must have been converted to a stop codon. So the arginine at the original position 57 has been converted into a stop codon. That's the only way we can get an incomplete protein. So again, we need to look at this genetic code table and see how we can get from an arginine to a stop codon. And you can see here that there's a lot of different ways. There's one stop codon. We could go from CGA to UGA, or we could go from AGA to UGA. So we have two possibilities, either one of those two. And I'll just indicate here which one it actually is. Speed this up. So it is a CGA. Has been converted into a UGA. Now, the second part of this question is that we have a second mutation that codes for a minor transfer RNA molecule, and it gives rise to a functional protein. And so the question is, what is the nature of this minor transfer RNA? And it codes for serine. So we have a second mutation here in a minor transfer RNA, and I'll draw it like this. We have a certain codon. It codes for serine, and we have to make a mutation in the transfer RNA of serine so it can recognize this UGA and read that as a serine rather than a stop. And the way we can do that is that if we had an AGU on the this transfer RNA as the anticodon, if that was coded for in the mutant 1B, this by the way is mutant 1, if this is now changed into a, from a, so this is gonna be changed from AGU to ACU. Okay, and now I think you can see that the ACU is able to recognize this UGA as a stop codon. So this is an example of a suppressor mutation, which is a secondary mutation that undoes the harmful effects of the first mutation. The first mutation is a nonsense. We change CGA to UGA, which is a stop. The second mutation changes an anticodon from AGU to ACU, and that recognizes that stop and allows the protein to be synthesized. It will now be a full-length protein, but with a serine here at position 57. And if that arginine was on the surface and didn't do anything essential, then it would be converted into something that was still functional.
How do you know it was a CGA originally instead of an AGA?
Are you coming down
Based on
tonight?